buonasera.
Good evening, ladies and gentlemen, and welcome to this session that I'm very happy and honored to introduce to you. The title evokes important and amazing features of our position in the world, of our condition in the world. And these features are disclosed by biology, an amazing discipline. We are fearfully and wonderfully made, the wonder of interdependence. And this has to do with any living organism and any living entity. So each organism becomes itself through what is other than itself. This is a key topic when it comes to the Being Alive exhibition. During this meeting edition, we decided to invite Professor Scott Gilbert, one of the scientists that contributed the most to the development of a new idea of a living organism. And he underscored the relational nature and character of living organisms. So I really want to welcome very warmly Scott Gilbert, and he's connected live with us from Portland, Oregon, and there it is morning. So a big round of applause for you, Scott. Thank you very much for being here with us. And the title of this very same session was suggested by him. And now I would like to introduce him to all of you here in Rimini or from anywhere you are following us from. And I would like to start from some personal notes. And I must say that I could appreciate him personally as author of a very important book, Developmental Biology, a book that is very well known. It was translated in many, many languages. It was published for the first time in 1985, and now it has reached its 12th edition by 2020. And after that, I had the privilege to meet him in person during a symposium that took place in San Marino, and it was organized with the support of Mating 2012. And that was entitled The Nature and Biological Evolution of Human Beings. It was a very demanding topic, and there were scientists and philosophers from all over the world. And Scott, at that time, really amazed me and other participants with the fascinating explanation of how symbiosis relations were able to have a deep influence on the history of life on Earth, and as a consequence also on contemporary life. And he's Emeritus Professor at Washington College in Pennsylvania and also at Helsinki University in Finland. Is one of the top leading experts in the world when it comes to developmental biology, but also when it comes to teaching biology. And because of these great merits, he got prestigious awards, the François Premier Medal of Collège de France, the Kowalewski Medal of the St. Petersburg Naturalist Society, and some original research work by him contributed to the foundation of the so-called Evo-Devo, and this is a meaningful contribution, a decisive one, to the evolutionary theory, together with other theories that were developed at a stunning speed from the mid-80s. Then Professor Gilbert is still contributing to an even better and deeper understanding of evolution, and as a consequence of all organisms, including ourselves, including also the ecological perspective within these general framework. And that opened up the way to the so-called Eco-Evo-Devo. It's a new field. And, well, there's so much happening, and he contributed with very high-level experimental studies and also with books that have become reference handbooks. All that would be far enough. But on top of that, the originality and creativity of his thought go beyond that, because he has a biology PhD, but also degrees in history of science, on religious topics, and also wrote important articles and books on the history of ideas, especially when it comes to biology. In one of these articles that was published on an international magazine that was curated for years by the Emeritus Association that I'm part of, Scott identified in Amazement a sort of primary experience that is, carry also many other things, that it constantly renews itself, also in embryology. And at the very heart, there is always somehow the show of the bodies of living creatures that form harmoniously from an undifferentiated set of cells. I'll talk about that article because there are many references already to Abraham Heschel, and this year we have a title taken from his works, Devoid of Wonder, We Remain Deaf to the Sublime, by the way. So, all that said, all these affinities, unexpected and amazing, my very first question to you, Scott, and I'm sure that you will start amazing us right from the start in the biological field. So my first question is, how is it possible for biology, that is a scientific sort of subject, to have to do with the sublime?
Thank you very much, and it's a pleasure. Great pleasure to be here. And we'll talk about this notion of being fearfully and wonderfully made. And it gives me the opportunity to talk about the wonder of interdependence. Now, the topic, if I can do this, of the seminar is Devoid of Wonder, We Remain Deaf to the Sublime. And that means the sublime is always there. It's just that we cannot hear it. And the sublime is different from what we consider the beautiful. The beautiful reaffirms reason. It's consonant. Whereas the sublime is something that can be fearful, powerful, even destructive. What the sublime affirms is that reason is not sufficient to apprehend reality. The sublime is wonder, but accompanied by a sense of terror, that this does not have to happen. And, oops, and something just happened here. Hello? Let's see. Try to get back. Let's see. I don't know what is happening. Can you see the slide? Okay, good. Sense of terror. Okay, so let me tell you something wonderful that may awaken us to the sublime, something which is terrifying, awesome, and possibly able to change our identity, something which in a way is monstrous. And that is that you are indeed fearfully and wonderfully made, but you're not who you think you are. You are not merely an individual. Often we think of ourselves as individuals, but you are not merely an individual. You are a biome. You are a collection of functionally dependent ecosystems containing hundreds of species. Now, usually, when we think about ourselves, we think of ourselves as a countable individual, and that's our anatomical individuality. Our body has one genome, and it integrates all the cells into a wholeness. There's also physiological individuality, that we have all these integrated organ systems which are to a common end. Then there's developmental individuality, that we are the products of the fertilized egg, that we have a fertilized egg made at fertilization, and then we are just the products of that egg. We have the notion of immune individuality. This is the individuality that if you were to put your skin onto me, I would reject your skin because it is foreign to me. That this is the rejection of others. It separates us from the world. Then there's genetic individuality, that the genome of my body differs from the genome of your body. And then there's evolutionary individuality, that the individual is that gets selected. Natural selection allows the survival of some individuals and not others. So these are the different ways that we've constituted individuality in biology. And what I am going to say is that they're all wrong. I'm going to talk about a notion called the holobiont, which is the animal or plant plus its persistent microbial communities. So we have here a cow. And when we think of a cow, we think of a female bovine animal that eats grass and turns it into milk. However, the cow cannot digest grass. There is no enzyme made in the genome of a cow which allows it to digest cellulose. Similarly, you have corals. We think of corals as individuals, but they are not. The coral contains an algae. And this algae is symbiotic such that it makes the food necessary for the coral to live. So the cow can't eat grass. It has symbionts in its gut that allow it to eat grass. The coral cannot make its own food. It has algae in it which can undergo photosynthesis to make the food for the coral. The termite here, shown at the bottom. The termite is supposed to eat wood, but the termite cannot digest wood without its symbionts helping it to digest the wood. So we have here the notion that the animal can't exist without its symbionts. And so when we look at anatomical individuality, we find that we are not these countable individuals that you see. We are more than that. We are ecosystems. We are biomes. 50% of the cells in our body are not from the fertilized egg. They are microbes that we collect from the moment of birth. We have about 160 microbial species per person. Each pore is a separate ecosystem. These animals, these microbes that live on us, have specific parts. Some live in our mouths, some live in our guts, some live in our skin. They live all over on every surface that's exposed to the air. So we are not only an individual anatomically, 50% of our cells come from microbes. How do we get these microbes? Well, some animals get them by what's called vertical transmission. They get them from their mother directly. They get them from the cell that's going to become the egg. Most mammals get them by horizontal transmission. So that here's a giraffe being born from its mother, and as it passes through the birth canal, as it goes to be born, it collects bacteria from the mother's vagina, from the mother's birth canal, from the mother's anus. It gets a huge number of bacteria. It gets, eventually, these will grow into about 8 million different genes in it, compared to the 22,000 different genes that we get from our parents. So one way of getting the bacteria is as we pass through the birth canal, our amnion breaks. We are now colonized as we pass through the canal. Another way of getting it is through feces. The koala bear actually mixes its milk with its feces and spreads it on the baby. Then another way is getting the bacteria through the environment. So the squid, the Bobtail squid of Hawaii, is born without a light organ. It needs its light organ to exist, but it doesn't have one. However, it absorbs bacteria from the environment, from the seawater. And when it absorbs the bacteria from the seawater, the bacteria help make the light organ, so that you have transmission vertically, directly from the mother, or you have it horizontally as the individual is being born or nurtured. Now, we have, as humans, a phenomenally interesting way, a really sublime way of getting bacteria. The bacteria in the mother's birth canal differs from the bacteria that's usually there if the mother is about to deliver the baby. The bacteria in the birth canal of a third trimester pregnant woman, a woman who's about to deliver the baby, differs from the normal bacteria. The hormones in the mother's body has actually selected certain bacteria. These bacteria help the mother with the pregnancy. They also are bacteria that you want the baby to have. And so the baby, once it's born, gets fed milk. And the milk has two types of nutrients in it. One type of nutrient is to feed the baby. That's kind of obvious. The other nutrients cannot be digested by the baby. It cannot be digested by any mammal. These are nutrients which are specifically for bifidobacteria, the bacteria that you want to be the first bacteria to colonize the gut, because they are the bacteria that allow the other bacteria to colonize the gut. So we get a different set of bacteria from the mother than the mother usually has. That goes to the baby, that goes to us, and then we're fed mother's milk, which actually helps the bacteria that we want in the body. So we have a way of transmitting those bacteria from one generation to the next. What do these bacteria do? They help the body. Our body is physiologically connected to the bacteria. In some insects shown on the left, the pathways for different amino acids are actually specified between 3 or 2 different types of organisms. So you have here Planococcus, which is the insect. In the insect is a bacteria called Tremblaya. In that bacteria is another bacteria called Morinella. And the pathway for phenylalanine begins in the bacteria, and then it goes into the bacteria, the bacterial symbiont, another bacteria. Then it goes back again. And then finally it becomes phenylalanine by the last step, which is done by the insect itself. In mammals, the microbes are essential for normal digestion, for normal neuroendocrine function, normal bone development, and normal immune function. The bacterial physiology and our physiology are linked together. This is critically important. Some diseases are caused not by the environment alone, but by not having certain bacteria. As Smith said in 2013, there's microbial host co-metabolism, that there's intimate communication between the symbiont microbes and the host. As much as 1/3 of the molecules in our blood have a microbial origin. So the circulatory system extends the impact of microbes throughout the body. I'll give one example. In our gut, we have microbes, and the microbes tell the gut cells, produced serotonin. The microbes make short-chain fatty acids, which tell our gut cells make serotonin, which is a hormone. The serotonin in our guts then act to mature the nervous system in our gut. Now, that nervous system in our gut is critically important. The nervous system in our gut is necessary for peristalsis. Peristalsis takes the food from our mouth and excretes it out our anus. Peristalsis is necessary for the movement of food through and eventually to exit our body. It allows us to defecate. So our symbionts are critical. There's even in Hebrew a prayer for a successful defecation. It's to show gratitude. And in a way, it's to show gratitude for our being. Blessed are you, Adonai, our God, king of the universe who formed mankind with wisdom. This wisdom is this sublime wisdom that allows us to be integrated together with other organisms. Let me continue by talking not only about anatomy and physiology, but about development. We develop with the symbionts. So anatomically and physiologically, we're holobionts, organisms composed of a consortium of interacting species. I now want to talk about development. And I have here a slide of the orchid, because the beauty of the orchid is due to the presence of a fungus that actually invades the orchid's eggs and supplies it with nutrients. The seed of an orchid is so small that it cannot develop. It doesn't have as many nutrients as it needs. The nutrients come when a fungus enters into the seed of the orchid. Now, I mentioned before the bobtail squid. The bobtail squid has a light organ, which it uses to it uses it to light up the seafloor to see prey and also to get rid of its own shadow, which other predators can sense. So it has a light organ that shines on the seafloor. But it's not born with a light organ. The light organ is created when it absorbs bacteria soon after it's born. It absorbs the bacteria, and the bacteria interact with the cells of its belly to actually create the light organ. In mammals, the blood vessels that take the blood and the food from the intestine into the rest of the body, those blood vessels are formed in part by bacteria. So here we have on the left blood vessels shown in green without microbes. It's a very poor vascular system. Not much food is getting to the outside. But when you add bacteria back, you get a beautifully made vascular system around the villus of the intestine, and this transports the food into the rest of the body. Now, how does this happen? This is really amazing. There is a type of bacteria, Bacteroides theta iota micron, and that bacteria is in the gut. It's part of the gut microbiome. And that bacteria produces compounds. And those compounds tell the cells of the gut, make a protein called angiogenin-4 and secrete that protein. And so the cells of our gut make angiogenin-4 and they secrete it. What angiogenin-4 does once it's outside the gut is it tells the cells around the gut, make capillaries. And so this bacteria is telling our gut to make a protein, which once it gets into the environment of the gut, tells the cells around the gut, make capillaries. The bacteria is inducing normal capillary formation in the mammalian gut. In fish, the gut cells tell the stem cells to grow. Without the microbes, the stem cells do not grow properly. So here in red, this is what you get if you do not have stem cells. This is what you get when you put in the bacteria. Normal development, the formation of capillaries, the formation of gut cells, depends on microbes. That's what we call normal. If you don't have the cells, if you don't have the bacteria, you get very little amount of angiogenin, for instance. So conventionally raised, you have tenfold more angiogenin mRNA than in mice without the bacteria. If you add Bacteroides back, you'll get the normal amount. So bacteria are needed for normal development. In fish again, a certain type of bacteria, Aromonas, is needed to get the proper number of pancreas cells developing. Without it, you don't have the normal number of pancreatic beta cells, you don't produce the normal amount of insulin, and you are prone to diabetes. The bacteria are needed for normal development. They are also needed for normal brain development. The gut bacteria are needed for normal gene expression in our brains, in the brains of mammals. Without the normal bacteria in our gut, we don't have the normal development of the brain. As a matter of fact, you don't have normal behaviors in the mice. Germ-free mice have behavioral anomalies. They have decreased sociability. So if you look at the control mice in red, germ-free mice without bacteria in blue, and germ-free mice given back bacteria in green, what you find is that most mice are social. But the mice that do not have the bacteria are asocial. They do not want to play with other mice. But if you add the bacteria back, they play with other mice. Similarly, for self-grooming, the mice that are germ-free groom themselves. They have self-grooming behavior. Instead of playing with others, they groom themselves. And you can, if you add bacteria, you get less self-grooming, more playing with others. So you have bacteria which are essential for normal brain development and for normal social behavior in other mammals such as mice, probably in humans as well. The bacteria influence us even before we are born because the bacteria that are in the mother's gut are able to produce compounds that go into the mother's blood, which go into the placenta, and which change gene expression in the developing embryo. It's found that certain microbes that are present in the mother are able to produce compounds that give the fetus, once it's born, the protection against metabolic syndrome, the protection against insulin resistance. So even before we're born, microbes are important in our development. They help produce our phenotype. So what I want to say, is development is a holobiont function. It's a team sport. Animals do not exist as independent entities. We become with the other. And that's important. In addition to evolution being competitive, it's also symbiotic. We become with the other. And the symbiosis is not merely between consenting adults. We actually develop using the symbionts. Yes.
Grazie Scott.
Thank you very much, Scott. What you said and that you told us and showed us is really enlightening and unveils a new dimension that is also somehow unsettling about our being living organisms. So we are holobionts, as you very well described, anatomically and physiologically. However, well, it doesn't stop there because we do not acquire our symbionts once we are adults because of interactions that occur from one point onwards. Instead, the somehow integration is more surprising because our embryo development, what is at the very basis of what we later become, well, unfolds according to this mode. So development biological procedures, occur according to a principle of becoming together with. This is a very powerful idea that has loads of repercussions. All that raises a question. So this compenetration of the different components of the holobionts raises this question. What is the role played by the immune system in all this? The immune system should somehow keep away foreign matters and protect ourselves from bacteria. How is it possible to have symbionts and have a functioning immune system at the same time?
Yes. Question. Is that in the next set of slides? Trying to keep the microbes out. What we now find is that symbionts help construct the immune system. And this slide here shows the gut-associated immune system normally with its activated B cells and T cells, the dome and follicle of the gut-associated immune system. But if you don't have the microbes, you don't have the gut-associated immune system. Germ-free animals have impaired immune functions. They don't have the correct number of T cells. They don't have the correct number of B cells. They don't have the correct tolerance. And what we find happening is that the original immune function of the body when it's born is to accept certain bacteria, and those bacteria help make the normal immune system. So what we find here, Fred Tauber says, from a philosophical perspective, the wavering ontological status of immunology's key concepts, self, individuality, and organism, highlights a science in transition. And immunology is a science in transition because it is trying to discover now how it is that the immune system is made by both foreign elements, the microbes, and host elements. So one of the things that's been found is that the immune system differs depending on whether the bacteria come from the mother or if they are gotten secondarily from the environment. If you take monkeys and you give them mother's milk, or you give them just a nutritional supplement, what you find is that the monkeys that receive the mother's milk have an almost totally different set of bacteria in their guts than the monkeys that were raised with just nutritional milk, but without the supplements of bacteria and food for the bacteria. What this means is that the monkeys get a different immune system. They get T helper cells if they've gotten mother's milk. They've gotten T helper cells that produce interleukin 17 and interleukin 22, which induces the gut cells to make proteins against Candida, against Salmonella, against Staphylococcus, in other words, against different bacteria. The bacteria that we take in from the mother protect us against a different set of bacteria which can harm us. Interestingly, the bacteria that harm us can also displace those bacteria. So the bacteria are, in a sense, using us to help defeat their competitors. It's a mutual form of defense. In helping us, the bacteria we get from our mother are helping themselves. And this is just amazing, that the bacteria help us form our immune system, and in so doing, protects that bacteria that helped us. So what you find in humans is that if you don't have a normal birth, if you have a cesarean birth, one is at risk for microbes for a different type of microbes, which include opportunistic pathogens, that you need these bacteria at birth. Different bacteria in infants born by C-section and who are not breastfed may actually cause higher morbidity than those who are breastfed and who do receive bacteria from their mothers. Also, it looks like certain bacteria from farms might have been critical in normal development to protect us from asthma. So there's all sorts of normal bacteria that we expect to make our immune system. So what I was learned what I was taught, and what I taught for several years, was that we had immune individuality and that the immune system was like an armed defense force protecting us from the outside environment. Actually, the immune system is more like a set of bouncers at a bar or passport control agents. They know who to let in and who to keep out, and they help establish symbiosis. This is a real new way of thinking about the immune system. Yes. So thanks for asking that question.
Grazie a te.
Now,
Grazie a te.
Thank you, Scott, for this answer that somehow invites us to open up our minds to new things that will certainly come up in this field. So the immune system works like an interface that is so delicate but sophisticated at the same time between the self and the other, has to be reconsidered as something that is even more sophisticated and even more refined, that considers processes that somehow still have to be fully investigated. And this is extremely fascinating. So time goes by rather quickly. We still have another 14 minutes. But I would like to ask you two questions, because have to do with our origin and nature. I start with the first question, and I hope to have the time to ask you the second one. The first question is, which are the amazing things that you told us about? What repercussions do they have for life on Earth? For the development of life on Earth, so when it comes to its origin. So somehow you made us understand that between the lines, but clearly, what is the impact? What was the impact of that on the evolution of life on Earth?
Okay, let's go with that one first. Lynn Margulis said, life did not take over the globe by combat, but by networking. In short, she said, I believe that most evolutionary novelties arose and still arise from symbiosis. And there are many ways that this happens, but let me discuss one very important way, and that's the symbionts can open up new pathways of evolution, because we're not talking about the evolution of just the individual that comes from the fertilized egg, but we're talking about a community which is interacting and can interact differently with its environment. I want to talk about how symbiosis facilitated the evolution of herbivores, plant eaters. Now, one thing you have to remember is plant eating came after carnivory. It's easy to digest animals. It's much harder to digest cellulose, plant material. So plant eaters came after animal eaters in the order of evolution. And I want to talk about the most commonly known herbivore, namely the cow. Now, the cow eats grass and grains. It eats plants, and it has a special stomach for this, and that stomach is called the rumen. The rumen makes this suborder of animals, the cattle, the goats, the sheep, the giraffes, the deer. They have rumens that allow them to digest grass and plants and leaves. The rumen contains bacteria, and that bacteria digest the plants. The animal alone cannot digest the plants, but the animal has a rumen that allows it to digest the plants. So the rumen contains cellulose-degrading bacteria, fungi, and protists that digest the cell walls into small sugars. Then there are the fermenting bacteria that will take these sugars, and they will turn these sugars into compounds such as short-chain fatty acids, which can go through the circulation and become energy sources for cells. It also has methanogenic archaea, which make this process more efficient, and it has detoxifying bacteria because many plants have poisons in them. and you need bacteria to get rid of those poisons. Now, what's really amazing is that you have this complex ecosystem, this stomach with all its bacteria, this rumen with all its bacteria. That rumen was made by the bacteria. Newborn calves have sterile rumens. They have no bacteria in them. However, within 1 to 2 days after birth, the bacteria that it got from its mother's birth canal have entered into the digestive system and enter into a small area which will become the rumen. And they stay there as long as the cow drinks its mother's milk. It doesn't do a thing until the cow starts eating grains or grasses. Then the grains and grasses enter the digestive system. These bacteria that are in this flap, which will become the rumen, start digesting the grain and the grass. And when the grains and the grasses are digested, they make short-chain fatty acids. And what do these short-chain fatty acids do? They cause the differentiation and the growth of the rumen. The rumen is caused by the bacteria. The bacteria digest the grain makes products. The products of the grain digestion are sufficient to cause the rumen to grow and to differentiate into the rumen, which will house more of these bacteria, which will allow the animal to get its nutrition. Butyric acid is the chemical that these bacteria make, and I can go through the slide, but I won't. But basically, the bacteria induce the activation of genes in the gut that make the rumen. So the rumen bacteria are present on day 2. They ferment the grain into butyric acid. The butyric acid makes the rumen. This is developmental symbiosis. Even if you were to add butyric acid into unweaned cows, you would get them to make a rumen. Now the bacterial community can ferment the grasses and grains into simple carbohydrates and allow the cow to grow. So the bacteria, the symbionts, have allowed a whole new evolutionary pathway for these animals. It allowed herbivory. It allowed the animal to digest plants. We find that microbes have scaffolded major evolutionary transitions. Or this is the hypothesis. The origins of multicellularity might have come through symbioses with microbes. The origins of meiosis may have come through symbioses with microbes. The origin of neurons may have come through symbioses with microbes. And the origin of the mammalian placenta and the uterine tissues may have come through symbioses with microbes. Microbes are critically important in allowing new developmental and evolutionary trajectories to come into being. So, we are fearfully and wonderfully made. In 1190, Maimonides, a physician and rabbi, said, "A pious man of my time would say that an angel of God had to enter the womb of a pregnant woman to mold the organs of the fetus. This would constitute a miracle. But how much more of a miracle would it be if God had so empowered matter to be able to create the organs of a fetus without having to employ an angel for each pregnancy?" Well, my job as a developmental biologist is to look at how matter can make embryos. And as Robin Wall Kimmerer said, doing science with awe and humility is a powerful set of reciprocity with the more than human world. Science can be a way of forming intimacy and respect with other species that is rivaled only by the observations of traditional knowledge holders. It can be a path to kinship. And what I had mentioned in the San Marino seminar was that nature can instill in us wonder. But wonder has a short half-life. One can't constantly be in wonder. And it becomes awe and curiosity. The English word wonder can mean I wonder, or it can mean the wonder of things. So you have awe and curiosity coming from wonder. From awe, we get wisdom and reverence, which leads to religion. From curiosity, we get the knowledge and questioning that becomes science and philosophy. And I'm saying that only when we understand nature, only when we have this continual source of wonder, can we generate the awe and curiosity needed to sustain religion and science. This is what makes things sublime. You have awe and curiosity together, that nature is a sense of the sublime. It is the evolutionary strategy that supports life on Earth. And I just will end with this notion of defamiliarization, astronomy, this Russian word, a technique for presenting to audience common things in an unfamiliar or strange way so that the audience can gain new perspectives and see the world differently. And this leads, I believe, to Heschel's notion of radical amazement. Awareness of the divine begins with wonder. The greatest hindrance to such awareness, what is keeping us deaf, is our adjustment to conventional notions, to mental cliches. Wonder is a source of radical amazement, the state of maladjustment to words and notions, which is therefore prerequisite for an authentic awareness of that which is. Our goal should be to live life in radical amazement, to get up in the morning and look at the world in a way that takes nothing for granted. Everything is phenomenal. Everything is incredible. Never treat life casually. To be spiritual is to be amazed. Thank you very much.
Grazie. La platea.
Thank you very much indeed. The audience that is here is really appreciating your speech with big round of applause. Thank you very much indeed for this amazing picture that you painted and for taking us to this journey through hidden and difficult passages unveiled by biology, and also for conveying somehow some of these radical wonder of which we're made of. And I have another question. Actually, I will keep it in stock and use it in a few days' time, because there will be the opportunity to ask Scott questions during a specific webinar that is going to occur on Thursday the 20th at 11 in the morning for you, Scott, and 8 in the evening for us. And also, please do enjoy our Being Alive exhibition that intertwines with many of the things that Scott mentioned. And I would like also to sincerely thank Scott. Thank you very much indeed, and thank you all for being with us. Thank you, Scott. Goodbye. Thank you very much. Bye.
days of hunger, won't you buy